Carbohydrates are sugar molecules. They are found in living organisms as both a form of energy storage and as a structural molecule. They are made up of carbon, hydrogen, and oxygen. Their general molecular formula is a multiple of CH2O. So for every one carbon, there are 2 hydrogens and 1 oxygen. Now this is because in a carbohydrate, almost all of our carbons are going to be linked to 2 hydrogens, a hydroxyl group, and another carbon. Our carbohydrate molecules are going to be made up of either a chain or a ring of carbons. So for example, glucose is made up of 6 carbons, 12 hydrogens, and 6 oxygens. The general structure of glucose is a 6-carbon chain. On one end, we'll have 2 hydrogens and a hydroxyl group, and each of those carbons is going to be linked to a hydroxyl group and a hydrogen, including the very last one. But that last one is going to be double bonded to an oxygen and a hydrogen. You might also see these in a ring structure. I'll show you those in just a minute. Carbohydrates are generally going to come from plants. So when we think about our diet, we think about things like wheat, grain, rice, and corn. The reason is that when plants take in sunlight energy through photosynthesis, they convert that energy into the energy of the chemical bonds into a carbohydrate molecule, usually glucose. And then plants will store that glucose in the form of cellulose or glucose, and it can be used as energy or it can be used as a structural molecule. Then, when other living organisms eat those plants, they take in the glucose, can use it in their cells to convert energy as well. Another name for a carbohydrate is a saccharide. You might have been familiar with that term. If not, that's okay. Our carbohydrates are going to be made up of individual units called monosaccharides. In other words, single sugars. These are also called simple sugars. So glucose is one example. Fructose is another that you might have heard of before. Those are all monosaccharides. The most common types of monosaccharides are 5 or 6 carbons. A few of them are really, really important in biological molecules. Ribose and deoxyribose, which have similar chemical formulas, are part of our nucleic acid molecules. In other words, part of our RNA and our DNA. Glucose is going to be the basis of almost all of the carbohydrates that we're going to talk about in our semester. Glucose makes up molecules such as starch, cellulose, and glycogen, which are all present in living organisms as structural or energy storage molecules. Your book is generally going to draw a monosaccharide as a ring structure. There's our ring structure of glucose. Sometimes they'll be drawn as a linear molecule, which I drew on the previous slide. In your ring structure, you always have an oxygen. At each of these points, you have a carbon molecule, and then coming off of your carbon molecule, you'll have your hydroxyl group and the other hydrogen that makes up that 4th bond in that carbon. If you remember from the previous slide, I drew a ring structure with a double bonded oxygen. That double bonded oxygen is actually the one that becomes part of the ring. Now, these molecules are most stable in their ring form, and usually if they're present in their linear form, they're going to fold into their ring form. These carbohydrate rings also have a carbon that sticks off of the top, and then all of our carbons are going to have a hydrogen and hydroxyl group. You'll notice that these molecules are numbered. That's not super important right now. I'm just going to point out that we can number our carbons, and that's to keep track of the carbons and to help us know which carbons are going to be involved in important bonds. They're always numbered starting from the oxygen and going clockwise. So our first carbon is the first one from the oxygen, second moving around clockwise, and so on. With the last one, the one that is a functional group, sticking up off the top. By linking two monosaccharides together, we can produce a disaccharide. In other words, a molecule made up of two individual sugar molecules. To produce a disaccharide, we're going to join these molecules by a dehydration reaction. Dehydration reactions can also be called condensation reactions, so you may see that in your textbook as well. The specific bond that results from the dehydration reaction of two monosaccharides is called a glycosidic bond, or a glycosidic linkage. So for example, a couple of disaccharides here, sucrose, maltose, and lactose. Sucrose is a disaccharide that is made up of glucose plus fructose to produce that disaccharide sucrose. So again, I'm going to draw our glucose molecule. I'm not going to draw all of our hydroxyl groups and our hydrogens because it's going to get a little bit busy. But I am going to draw the ones on this side. The reason is that we need to find a water molecule to link it to fructose. Again, I'm not going to draw all of our hydrogens and hydroxyl groups because it's going to get messy in there. But I am going to draw the ones that are going to be involved in our chemical reaction. So we need to find we need to find a water molecule. So we need to find an H2O. Now, the way that a glycosidic linkage is going to work is that we are going to produce a molecule that is connected by an oxygen in the center. So we actually need to find water plus an extra oxygen. So if we look at our components here, we've got two hydroxyl groups. In a glycosidic linkage, we are going to pull out water, and we should be left with this oxygen that's forming the bond in the center. So we're going to pull out water. What's going to happen is that one hydrogen is going to combine with a hydroxyl group, and we're going to be left with that oxygen there. Combine, linked by this oxygen in the center. Now that's a little messy, so we are going to move on, and I'll show you another picture and explain how our glycosidic linkage is formed. So we start with glucose and fructose. We're going to perform a dehydration reaction between the hydroxyl groups on both of those molecules. We're going to pull out water, and what we are left with is one of those oxygen molecules. That's going to be this guy here. That oxygen molecule is what is linking that glucose and that fructose. So again, that oxygen bond in between those two monosaccharides is your glycosidic bond or your glycosidic linkage. So when this happens, we are left with sucrose plus our resulting water. Remember that in all of our dehydration reactions, we are going to remove 1 water for every bond that we form. So we are forming a bond between your glucose and your fructose. Now we can also break our glycosidic linkage by hydrolysis. Remember that hydrolysis is the process of using water to break the bonds. So we can do the same thing and go from our sucrose down to our glucose and our fructose. In cells, most of our sugar is not going to be in the form of a monosaccharide and a disaccharide. Monosaccharides are absolutely used, and we'll see later in the semester how individual glucose molecules are actually used to produce our energy molecule ATP. But in living organisms, we often find our carbohydrates in the form of polysaccharides. Polysaccharides are long chains of monosaccharides that have 2 common functions in living cells. One of them is energy storage. So plants store their monosaccharides as the polysaccharide starch. You'll notice that starch is a chain of glucose molecules linked together by a glycosidic linkage. Now, starch is a single chain. It doesn't branch very much. It's not a very compact molecule, which is why plants can use starch. They're not going anywhere. They don't have to carry it around. It's okay if it's bulky. In animals, like my cat here, that's Oreo, we use polysaccharides in the form of glycogen for some quick energy storage. Glycogen is really similar to starch in that it is a series of glucose molecules, but it's a little bit different in that it actually can branch. So that branching makes it a little bit more compact. Either way, plants or animals can take their respective polysaccharide, break off glucose as they need it for energy, and use it to produce ATP. Polysaccharides also have a structural function in living organisms. The most common structural molecule is cellulose. Cellulose forms the cell wall in plants. Now, cellulose is important because cellulose is what gives plants their vertical structure. It's what supports them against gravity. They don't have bones like animals do, and so they need something to support their outer structure and hold them up. Cellulose is really similar to starch and glycogen, except for one key feature. It's still made up of monomers of glucose. It still has that glycosidic linkage. But what's different is that your glucose molecules are lined up in opposite directions. And what we get is this alternating glycosidic linkage. When that happens, your cellulose chains can lay down next to one another and actually form linkages between other cellulose chains and themselves. This is what causes the ability to provide structural strength. Starch and glycogen don't cross-link, so they are just found in chains, whereas the cell wall acts almost more like a net or a fabric. Couple other polysaccharides that are important structural molecules. Peptidoglycans are found in the cell walls of bacteria. Chitin is found in the outer structure, the exoskeleton of arthropods. So insects, crustaceans, organisms like that, where their structural support is on the outside and not their inside.